Gene therapies are an amazing branch of medicine that show enormous potential. Many medical experts and bioethicists believe that the future of medicine will revolve around custom gene therapies tailored for the genome and genetic health of individual patients. Today's news story should be an eye-opening example of the potential utility of gene therapies and research into the medicine of genetics. Researchers from the University of Pittsburgh, the University of Cambridge, and the Institut de la Vision in Paris have recently written about a Phase 3 clinical trial that used an injected gene therapy to treat 37 patients with Leber hereditary optic neuropathy, or LHON. The LHON disorder is a maternally inherited mitochondrial genetic disease that causes retinal ganglion cells to degenerate and fall apart. As these ganglion cells link sensory input from the eyes to the optic nerve leading into the brain, LHON leads to a rapid deterioration in vision that ultimately leads to near or complete blindness. Less than 1 in 5 patients ever show any signs of recovery, and treatment options are very limited. Among the 45 different mutations that have been linked to LHON, many patients have a specific mutation in the MTND4 gene, which codes for the NADH ubiquinone oxidoreductase chain 4 protein. The mutation occurs in the 340th codon of the protein, turning an important arginine amino acid into a histidine. This mutation alters the protein's function, leading to impaired ion conduction across the mitochondrial membrane. This has all manner of downstream consequences, including the inability to manage ion gradients and impaired function of the electron transport chain and reduced ATP production. The researchers developed a viral vector called RAAV2/2-ND4, that's a mouthful, which uses viral protein mechanisms to integrate a designed gene into the cells of the eye. This viral vector was injected into the vitreous cavity in the back of one eye, in 37 patients who had experienced vision loss for 6 months to a year due to LHON. The other eye received a sham injection, which is a research control technique that simulates the injection process but doesn't actually inject anything biologically significant, anything beyond like physiological saline or some neutral solution like that. Now, one of the lead investigators in the trial, Dr. Patrick U.Y. Mann from Cambridge, described the process, saying, quote, "By replacing the defective MTND4 gene, this treatment rescues the retinal ganglion cells from the destructive effects of the M.1177HEA mutation, preserving function and improving the patient's visual prognosis." Unquote. About the results of the study, Dr. Yue Mann said the outcomes could be, quote, life-changing, unquote, and also said, quote, "We expected vision to improve in the eyes treated with the gene therapy vector only. Rather unexpectedly, both eyes improved for 78% of patients in the trial following the same trajectory over 2 years of follow-up," unquote. To demonstrate how impressive the improvement in patient vision was, think of the vision test at the eye doctors, where they have you read lines of letters printed in increasingly small font. This viral vector injection, this gene therapy, was able to improve visual acuity in both eyes to such a degree that the patients could read 3 more lines on the chart than they could before the treatment. 3 more lines, that is a massive improvement. And the researchers didn't stop here. After observing these unexpected results where the improvements were seen in both eyes, they wanted to know why and how this happened. So they conducted a similar study in model primates, and after 3 months, they examined the primates' brain and eye tissues to try to identify any viral vector DNA. And they did, identifying traces of their injected gene therapy in the retina, in the optic nerve, and in the anterior segment of the untreated eye. So, in more general terms, it would appear that the gene therapy, when injected into one eye, can travel through the optic nerve, where it presumably crosses the optic chiasm and spreads to the optic nerve of the untreated eye. And if you're not familiar with the optic chiasm, it's a point between your eyeballs and where the optic nerve reaches the brain, where the optic nerves actually cross over. So the optic nerve coming from your left eye crosses over and connects on the right side of your brain, and the optic nerve from your right eye crosses over and connects to the left side of your brain. And that point where they cross over is called the optic chiasm. this appears to be the junction through which the gene therapy travels to improve both eyes. Now, this development is pretty amazing because it suggests that more medications might be able to be administered in this way. Dr. Yue Man said, "Saving sight with gene therapy is now a reality. The treatment has been shown to be safe, and we are currently exploring the optimal therapeutic window," unquote. Another lead researcher, Dr. Jose Alain Sahel from the University of Pittsburgh, said, quote, "Our approach isn't just limited to vision restoration. Other mitochondrial diseases could be treated using the same technology," unquote. So all in all, this is super exciting. This is a really amazing gene therapy, and when they used it thinking they would get a certain result, they actually got twice the results. These amazing unexpected results where both eyes improved from a single injection in one eye. And the fact that this technology could potentially be used to treat a whole host of other mitochondrial diseases, that's really exciting. And if you're a patient who has one of these mitochondrial diseases, technology like this can be a source of hope for a future treatment or even a cure.